being here. Just a few starting comments before we turn it over to you. One thing we're going to do might seem a little bit awkward to you guys, that it's very important anytime you're presenting to any group, whether it's a lecture hall full of 500 people, or whether it's 3 people, or whether it's one-on-one, is to try to get a handle on who is your audience, where's their knowledge level, what do they know about this stuff, right? So you know how to deliver your message. So you know whether to aim here, or aim here. So we're going to formally introduce ourselves to you, and give you just a sentence or two about where our understanding and knowledge might be that may help you adjust as you go through as to how you want to present some technical items. But before we go there, I want to just remind you that any commentary, criticisms, critiques, feedback we give you is the only reason we do that, the only reason they have this process is so that 2 years from now, you guys have a great product. You've done a lot to now, and you're kind of on that launch pad. You kind of get ready to spring from background preparation, gathering information, to actually doing something. And we want to make sure that you have a good enough understanding of where you are, so that when you launch it into action, it will be a very efficient use of your energy, very high probability of successful outcome. And everything we give you is just to make sure that your thesis 2 years from now is great. Okay, this is, even though it's a nice event, a nice product, you guys put a lot of effort into it, it really doesn't matter at all. Okay, it's a stepping stone towards 2 years from now, a continuous process that we're launching out onto. So just take it for what it's worth, okay, and take all our comments and use them. Okay, that being said, I'm Dr. Frank Cole. I'm director of the Gemstone program. Regarding this topic, I have a solid fundamental understanding of biology and biological systems. Cancer cell growth and cell transfer, et cetera, like that is on the very periphery of my understanding. So that's where I kind of land. So you'll know that going forward as you talk about your topic. Kristen, you want to go?
Yes, hi, I'm Dr. Kristen Sandel. I'm the Associate Director of the Gemstone program. I haven't taken biology in 20 years, but I did have skin cancer. So I'm excited about your product, and I think the Mohs piece in there hit home with me, because I hope no one has to have that ever again. It was not a fun surgery.
Either one.
My name is David Moser. I'm a professor in cell biology and molecular genetics. In the spring semester now, I teach immunology to undergraduates, and in the fall semester, I teach immunology to graduate students. So I know a little bit about cytotoxic T cells.
More than I do. My name is Jing Xiao, and I'm an assistant professor in animal science. I'm teaching physiology, in addition to in the spring, and teaching immunology, comparative vertebrate immunology in the fall. And I think you guys choose a very complicated model, So, but it's not because you like that, but we don't. I think it's I'm amazed how fast you guys learning through this. Even sometimes I'm not very kind of in good mood, but you guys follow that very well. So I think you guys learn very quickly. So I'm impressed by that. So I'm looking forward to continue to learning from you guys.
Okay.
I'm Eileen Harrington. I'm the Health and Life Sciences librarian. I do have an undergrad degree in biology and environmental studies, as well as a master's in environmental studies, but my biology is more focused on conservation, biology, and ecology. So some of this I remember from my biology courses, but not as extensive as others.
Okay, that's who we are, so tell us who you are.
Okay, I'm Kira Egan. I major in cell biology and genetics with a minor in Italian, and I'm a sophomore on Team Basic. What else do we explain?
No, that's it. You can just start.
Okay.
However you want to start your presentation.
And I'm Jordan Litter. I'm actually a business marketing major, but I'm also doing pre-veterinary studies, so I have a pretty solid biology background.
I'm Solo Beg. I'm a biochem major and sophomore here. And before we begin, I just wanted to let you know that I will be leaving early because I have lab after this, so directly after the presentation I'll slip out. But I apologize for any inconvenience that may cause.
Okay,
Now,
that's fine.
Jordan?
Class is class.
All right. Thank you everyone for coming today. As I said before, I'm Jordan Litter. So Kira and myself will be presenting Team Basic's proposal today. So our research question is going to focus on the effect of neutralizing the extracellular pH in melanoma cells and the effect this will have on the efficacy of adoptive cell transfer therapy. Specifically, we're going to be looking at bicarbonate, and we hypothesize that by treating melanoma cells with bicarbonate, we will improve the efficacy of adoptive cell transfer therapy because we will be increasing the access that T lymphocytes have to the tumor. So our diagram up here kind of explains our project overall. We're going to be taking the inhibition that the extracellular pH causes on adoptive cell transfer therapy, and we're going to try to buffer it with the bicarbonate, and then combine these two things for more effective melanoma treatment. Kind of as an overview, I'm going to start out by talking about the basics behind melanoma, and then Sybil will take over and get into ACT and pH and bicarbonate. Kira will then go forward with our research question and methodology a little bit more in depth. So the incidence rate for melanoma has nearly doubled in the last couple of years, going from about 36,000 to 61,000 incidences. Obviously, this means that the current treatments are not effective enough, and there's no long-term effective treatment. Melanoma is often caused by thymine dimers from sunlight and UV radiation in the cancer cells in the DNA. And this can cause cancer to form through mutations. It then can metastasize to other parts of the body, which is what makes it so deadly, because any epidermal tissue in the body can have a melanoma tumor, so such as cells in the urinary tract. And melanoma also is very much like other cancers in that it has this extracellular pH that's more acidic than a normal cell, setting it apart from healthy cells and helping to inhibit the immune system from reaching it. Some current treatments that have been out there for both melanoma and other cancer cells are surgery, chemotherapy, and radiation therapy. Now these are, while they're semi-effective, they also harm a lot of healthy normal cells, and this can cause other problems in the body, such as through chemotherapy harming blood cells and hair loss cells through attacking cells that multiply rapidly. It can lead to anemia and hair loss. Right now, the future of cancer treatment is leaning towards targeted therapies because they have less of an effect on these healthy cells. Some current ones that are out there focus on the unique attributes of cancer, such as the protein pumps that they use for increased glycolysis, as well as the extracellular pH and the fact that cancer cells multiply rapidly. So this is the future of cancer treatment at the moment, which is why we've chosen adoptive cell transfer therapy, because it is one of the most effective therapies on melanoma. So Sybil will now take over and explain that a little more in detail.
Thank you. Alright, so as Jordan mentioned, adoptive cell transfer therapy is one of the newer targeted treatments out there for melanoma right now. What this therapy does is it takes T lymphocytes out of a biopsy of the patient's tumor, enhances and multiplies them with proteins to make them stronger and more effective ex vivo, and then reinserts them back into the tumor site. The T lymphocytes that are in question here are called tumor-infiltrating lymphocytes, and what that means is that they essentially have an affinity for the tumor already, so ACT is more of a natural treatment for the body. On its own, ACT is relatively effective, so it's usually combined with other treatments to enhance its impact. And one of the main reasons that ACT isn't as effective on its own is because it does require a large number of cells to be completed. This means that a lot of T lymphocytes need to be reprogrammed and introduced into the tumor, so it has some impact on the cell. In addition to this large number of cells, ACT also takes a very long time to complete, about 4 to 6 weeks, which isn't ideal for a cancer treatment, and these two factors combined make it a very expensive treatment. In addition to these technical problems, there are also some physical side effects listed here that are basically either pro-inflammatory responses, high fevers, or some minor functional failure within the body. Now, a major reason why ACT has all these problems is because of the acidic microenvironment of the tumor. This acidic environment tends to have a suppressive effect on the immune system and on ACT. In one study done on human peripheral blood cells against an erythroblastoid cell line, they reduced the pH from 7.2 to 7.0, which is a slight reduction, and they saw two things happen. One, the cytotoxic effect of natural killer cells was significantly inhibited in this acidic environment, and lymphokan-activated killer cells weren't even being produced in this environment. So as you can see, the acidic environment tends to have a detrimental effect on immune cells. Another study was done on the major histocompatibility complex, which is a group of proteins found on the immune cell surface, and it sends out signals and recognizes foreign substances within the body. So in an acidic environment of a pH of about 5.8, this MHC complex was inhibited and it wasn't recognizing the tumor. So the tumor was essentially invisible to the immune system. So from these couple of examples, you can see that the immune system and ACT don't work well in an acidic environment, and so this environment needs to be overcome. One way to do that is through manipulating the extracellular pH. And by neutralizing these pHs, or this pH, you can solve a lot of technical problems with ACT. One being the large number of cells required. In the acidic environment, a number of cells can't reach the tumor site, so you need to make more just so some of them can reach it. But if the acidic environment is neutralized, more cells will be reaching the tumor site, and they will be reaching them faster, so you solve the issue of time. And if you solve these two factors, you solve the issue of expense and cost for the treatment. So one way to neutralize this environment is through the use of a basic buffer. One we've been studying is bicarbonate, which has been orally administered to mice with breast cancer, and it's been extremely successful in reducing the metastases of that cell line. So what we want to see is that it also reduces the metastases or neutralizes the pH in melanoma skin cancer as well. So now Kira will tell you more about our research question and methodology.
So to go into the more specifics of the ACT that we will be doing, as Sue will explain this part, specifically the enhancement, we will be adding 2 proteins to the tumor-infiltrating lymphocytes to enhance them. And these 2 what they do is the first one, interleukin-12, this one right here, enhances the tumor-infiltrating lymphocytes to fight the tumor off better. The immune response is improved with these. And then B7 stimulates the growth of these in ex vivo before we introduce them into the body. So these 2 tumors were increasing the efficacy of their immune response, and we're also making a lot more of them. Specifically with the interleukin-12, what it does is it stimulates the growth of the MHC complex on the tumor surface, and then it causes tumor suppressor genes to be expressed as well. And with these 2 things, we hope to have a more effective ACT therapy. And the B7 is very important because it stimulates all the growth, it reduces the amount of time needed for the ex vivo expansion. So in front of us, our research focus and our question is, what effect does neutralizing this acidic extracellular environment have on increasing the efficacy of adoptive cell transfer therapy? And in front of this question, our hypothesis is that direct treatment of sodium bicarbonate will neutralize the extracellular pH of the melanoma tumor cells and therefore increase the efficacy of adoptive cell transfer therapy. And the way we will be doing this is in 2 phases. The first phase will be a proof of concept phase to understand the relationship of bicarbonate and melanoma tumors, and that'll be in vitro. And the second will be in vivo in mice to understand the relationship between bicarbonate and ACT in a mouse model. So our first objective, our hypothesis, is that the treatment of sodium bicarbonate to melanoma cells in vitro will increase the extracellular pH of the tumor microenvironment. And the way we will be doing this is we will have 10 groups of melanoma in vitro, and we will be treating them with bicarbonate with a range from 0 to 250 micromolars of bicarbonate. And this range has been found in previous studies to be effective on breast cancer treatment, so that's why we're using it. And from this, we will be testing these things, and the 2 most important things are the tumor growth and the extracellular pH. So from this, we hope to find that there is a relationship and that it does increase the extracellular pH, and also that there's an optimal dosage that we can find in treating the melanoma cells. And this optimal dosage we hope to be using in our mouse model. So for objective 2, our hypothesis is that the treatment of sodium bicarbonate to melanoma tumor cells will increase the efficacy of adoptive cell transfer therapy on the mice. The way we will be doing this is we will have 3 groups. The first group will be mice with melanoma only treated with bicarbonate. The second group will be mice with melanoma treated with both bicarbonate and adoptive cell transfer therapy. And then the third group will be mice with melanoma only treated with adoptive cell therapy. And from these 3 groups, we hopefully understand the relationship between bicarbonate and adoptive cell transfer therapy. So our expected results from these 2 phases is the first, we hope to see an increased extracellular pH in the melanoma from bicarbonate treatment, and also to find an optimal dosage of bicarbonate. And our second in vivo study, we hope to see increased access for the T lymphocytes to the tumor, and then therefore an improved adoptive cell transfer therapy. And now Jordan will explain our timeline.
Okay, so a brief overview of our timeline. This spring we want to submit our ICRIC proposal, begin applying for grants, and begin our in vitro experiments. So that come next fall of our junior year, we can finish the in vitro data collection and begin in vivo and be prepared for junior colloquium. And then in the spring of our junior year, we want to finish all the data collection and begin our data analysis and our thesis. So that come the fall of our senior year, we can complete all the data analysis and our thesis and be prepared to defend our thesis come the fall of our senior year. Just to wrap everything up, once again, our hypothesis and the design of our experiment is to take the effect that pH has on adoptive cell transfer therapy and try to neutralize it with bicarbonate to come up with a more effective treatment for melanoma. This is taking two of the most promising research areas right now, immunotherapy and the manipulation of pH, to combine them into a more effective cancer treatment. We're going to use both in vitro and in vivo testing to make sure that we have the most validated data possible. And we hope that in the future this can be applied to other cancer types as well outside of melanoma, so that it has a further reach. Thank you again for your time, and we're now ready for any questions. We also have our reference page right there as well.
All right, good job. Nice
Thank you
present.
guys for coming. I'm sorry I have to.
We'll just stand, skip out. Nice presentation, good job. Appreciate it. Our tradition is we start with our external expert
Oh, good.
to do the first round of bloodletting. So, and then Jinka will move over to you, and then we'll follow up at the end. So, go right ahead. Anything's fair game.
So, I appreciate that you guys have not had immunology, but I'd like to get a description from one of you or all of you what this process of cytotoxicity involves, just to see how much you've learned about this. Does anyone want to take that and sort of explain? For example, if you were to draw two circles on the board here and I called one of them a cancer cell and one of them a cytotoxic T lymphocyte, what molecules are on each of these cells that mediate this cytotoxicity? Is that, you know, did you review any of that in your readings?
I can do it, or you can do it.
You can use it.
You can all do it
Do you have
together. You
markers
can
that I
help
could
each
use?
other.
Because if I have a marker, I can.
Let me go get some.
Oh yeah, that's a good point. All right.
She just likes using the board.
I'm expecting tough question from the doctor.
And
Don't
we're
be
very
panicked.
open to correction, just to let you know.
It's afraid like, what about CD 84/7? Like,
There you go. Whilst with your
Yes,
art.
two colors, this
That
is
was gonna be
beautiful.
my second question.
Thank you for the reminder.
So you have all this, actually, the way I've been doing it. The tumor is a little messed up, it's a tumor. And then you have the T cell, okay? So on the tumor is the MHC. It's explained, and then the T cell has a receptor. And what maybe you can clarify is, I know this links to this through these two, but sometimes there's a link here, and sometimes there's not. We were talking about it last night, we were understanding either it links or not through this. Like, without this they can link, and with it they can link as well. So, that's what I understand about that.
Do you want to add a little bit there about what's
yeah,
inside?
it's like, you have interleukin, like, you're talking about interleukin 12, and what interleukin 12 is, it finds the receptor on the T cell, but that doesn't aid it in its bonding to MHC, the major histocompatibility complex. It pretty much just allows it produces more interferon gamma, which then causes the T cell to produce more MHC, so that more general T cell receptors can connect to the MHC complexes.
Yes.
So then...
Do you want me to draw it? No, like,
I mean, sure.
so we have IL-12 that we're adding, and then we have B7. IL-12 is a receptor on this, and there's a tumor that goes, there's a protein that goes here. This protein activates a cascade of events that produce gamma, interferon gamma. Interferon gamma, we'll just call this gamma. And this interferon gamma is released, gets into the tumor, and causes two things. Causes more expression of the major histocompatibility, this guy, and then gets into the genome and causes the tumor expressor genes to be expressed. And the
Oh.
B7,
Dr. Moses' question is, how did cell kill?
how does it kill?
Yeah, so
Oh, okay, then it very much disrupts the cellular membrane for the tumor cell.
do you remember what's that?
It just causes necrosis, the tumor necrosis factor. I mean,
This linkage causes, not death, but
What punched the
Yeah.
holes?
like
Oh,
apoptosis.
It goes boom.
Yeah.
Yeah.
They got the concept.
I know exactly what you're talking about, I
Yeah.
just can't remember.
So who wants to help her? What's the little red glob?
Yeah. That's interleukin 12.
That is
It's a
interleukin
protein.
12. And so what's the Y that you wrote attached to that glob?
The interleukin 12 receptor.
Right, okay, good, yeah.
But that was my question in studying it. Is interleukin 12 just the protein or the complex of the protein and the receptor combined?
No, interleukin 12 is the cytokine,
Okay.
so it's the protein
So it has its
that
own
binds to
receptor.
cytokine receptors on that
Okay.
cell. So that's how you're gonna stimulate that cell.
Mhm.
Anyone want to help her with the MHC? Should there be any globs on MHC?
There's, yeah, you taught this to us.
No, that's the TCR.
Like this,
Yeah, okay,
and then, I don't know what's the name, but something else helps this binding to it,
okay.
right?
Yeah.
But what's the, in the middle of the Y, between the MHC class 1 and the receptor? What is the specific? How, we talk about the killing is specific to tumor, right?
Yeah.
Is that just bind to MHC class 1? What is the conformation determination on that? How the specificity is determined? Wow. They're stressed.
Anybody can help at any
Specific antigen
time.
expressed by the tumor cell?
A whole bunch of specific
Yeah.
antigens
Yeah.
expressed by the tumor cell.
Yeah.
That's right. So if I were drawing this, I would have drawn a little glob here.
Yeah.
Yeah, see,
Just sitting in that MHC.
Oh, yeah.
That's really what the
That's-
T cell,
okay.
what the T cell recognizes.
Yeah.
okay.
Now, what tumor cell are you guys going to use?
B16 melanoma.
B16 melanoma, does it have any more of a name than that?
B16 ova?
What's the ova? Anybody know?
Mm.
So this happens to be a melanoma cell that's engineered,
Mhm.
so it has a specific antigen, so you can predict
Okay.
which of the tumor antigens are important. In this case, it happens to be really an uninteresting antigen, ovalbumin.
Mm.
But it's an antigen that we have plenty of around, and it's an antigen that we have a cytotoxic T cell that specifically recognizes. So a lot of immunologists use this because you know what this interaction is going to be. And it's going to be an OT1 T cell that is transgenic because it recognizes that ovalbumin peptide that's sitting in the MHC molecule. So that's why this, drawing a little blob there, that's why that blob is so important. Because it has to
The
be,
specificity.
you have to have some degree of specificity. So you could take, so you can't really take T cells out of a mouse or a person and know what those T cells are going to recognize on that tumor cell. But you can take an OT1 T cell that immunologists grow up in the lab all the time and add it to your melanoma cell, and that T cell will kill that melanoma cell because it recognizes that ova. And so it's a really elegant and simple model for you to use to actually look at the killing. But you guys don't take advantage of that model. And I think that's one of the weaknesses maybe of your proposal. It would have been a little bit more elegant if you would have added a middle aim to actually see, can you kill your tumor cells in a test tube just by adding a T cell with the tumor cell and seeing if you can kill it, and then seeing what happens if you start changing this pH.
That's great.
So a second in vitro,
So a second aim.
in vitro.
So right now you have one aim that says check the pH effects on killing the tumor cell, and the second aim is really the end. Take the tumors out of take the T cells out of the tumor and ask, will they work? Maybe that's too big of a jump. Maybe you should have had a little aim in the middle to see if we do this in the test tube, does it work, and how does the pH work? And then because right now making this huge jump, it's going to be hard. If it doesn't work at the end of your next 2 years, it doesn't work, you're really not going to know why it didn't work.
Mm.
You won't know whether the bicarb didn't get to the tumor. You won't know if the bicarb didn't make the tumor more susceptible, or you don't know if the T cell doesn't work better in the presence of bicarb. But if you add that middle aim, maybe you'll be able to have some answers to that question.
It's definitely a great suggestion.
Yeah, absolutely.
At one point, we were going to do, before we completely understood ACT, we were planning on doing it in vitro. And once we realized you can't do it in vitro, there's no tumor environment to do that with, and full immune system to work with, we discarded that idea, and I don't think we ever really revisited that, so thank you, that's a good work.
Excellent, and great art.
Yeah, that's good. That's why
I was surprised you guys get Dr. Moser to serve on the committee. That's really great addition to us and stuff, so.
Very good.
Very good.
So, you want to continue, or is that enough?
Wait for the second one?
Yeah, I'll wait for the second round.
Okay, go right ahead.
Yep, so, that's very good, that's very good. And the presentation wise, I was pretty impressed by, compared with last time and stuff, so you guys worked very hard on that, so. And you make the connections very well, so the justification, stuff, and I think the technical part is the details, you still need to work on that, such as you put the, in vitro and molecules B7, IO12, so is that just put those two, that's enough? It's, you're missing antigen, there. So because if you don't have antigen, you always have this figure as Dr. Moser suggested, so in your mind, so how the cytotoxic T cell respond? What do they require? As the first one, they need to require, the recognition of the antigen specific, the antigen on the tumor. So how you do that? Actually, in their your system, we coat that on the plates, on the dish, and then put it on the top, and they have automatic recognition on the MHC class 1 presented peptide from over, stuff. And in addition to that, you have B7, you have IO12. That's how the system is working. Yeah, so something like that. So for the acidic environment, so you say you can visualize, so you test in vitro to see how the pH can be changed by bicarbonate. So will that information be, translational, or informative for the in vivo? Because if you see pH, 6.8 is good, you can change that by a certain concentration of the bicarbonate. How can you use that information for in vivo? Is that determining how much you can inject in, how you do that?
Yeah, we first want to do the in vitro just to determine whether or not the bicarbonate actually will change the pH, because the studies that we read all focused on breast cancer with that. So we want to confirm that it actually works in melanoma before we start using the mice models. So yeah, based on how much it can change, based on how much bicarbonate we need to use to make that change, that'll go into our in vivo study, kind of deciding how much bicarbonate we're going to use. And ideally, part of the study is that we can use less bicarbonate and less of the adoptive cell transfer therapy than conventional treatments do right now, as that limits any side effects that either treatment might have. So ideally, we would probably start with what we think the optimal dose will be based on our in vitro, and then see if we can back off that a little bit and if it's still effective or not. Kind of play around with the levels to figure out where an effective level of bicarbonate would be without having an excess use of it.
Yeah, that's good. And another question is, the setting of the in vitro, it's very nice, because in my understanding, it's kind of a set up as a control. So if you change the pH, will that cause any effect on melanoma, which is not supposed to, right? So my question, you can simply add one more aspect to that in vitro one, because you can ask the question, if you expose to the pH, low pH, or kind of higher pH, will that intracellular pH also be changed for the specific cancer? Because it will be very hard to do for the in vivo, but in vitro you have a simple factor, right? You raise the pH in the actual cellular environment. Will that for 2 hours and 2 days, will that affect the pH inside the cells? Stuff, because before was not clear about that. So will that happen? Because the change of the extracellular and intracellular pH is kind of different questions. So you can dissect it very well in your system for the in vitro.
I'd like to follow up on that a little bit, if I may jump in here. You're going to rely on the published literature on breast cancer and the pH manipulation with the bicarbonate to determine what ballpark or range you're going to begin. Are you confident that that range is the responsive range for these different cancer cells, or could it be 10x, 100x, 1000x, more or less than that range? I hate to see you set up a whole study where you were not within the proper range because you're referencing a different cell type as your concentration reference. How confident are you in that?
That's a very good point, and we did try to look for more literature that involved bicarbonate with melanoma directly, and we did have trouble finding it. So that is kind of a risk of our in vitro study. Luckily, by doing it in vitro, we're going to be saving a little more money than we would be doing it in vivo. But we will have to definitely sit down, and I don't think we've really done that yet, play around with what we want our dosages to be to start with, and maybe have a couple within that range, and then one that's like a lot less and one that's a lot more, just so we can kind of get an idea of what range we should focus on for our actual experiment. That is a really good point that we have to think about.
Because if you're going to take a leap from in vitro work, which sounds like there's at least the in vitro studies you propose and the new intermediate level, it sounds like expanded intermediate in vitro level, making the leap to an in vivo project is a huge leap, and you're going to have very, very, so few number of individuals to work with. You don't want to be guessing at anything like rate, and you want to have that just nailed down. If you might think about it, if this new suggestions you've received really expands your in vitro testing and adds many different elements to in vitro testing, you may consider not going to in vivo studies. You may have your hands full of just doing the in vitro work and nail down the bicarbonate rate, nail down this why question that was brought up before. And if you nail all that down, that's a wonderful contribution. Without taking the risk of, maybe it's going to work when I plug it into the mice, maybe it's not. So just an observation from some experience.
I think that's very helpful. I think it's definitely a good idea for us to really make sure in vitro is as solid as it can be and prove our hypothesis as much as it can before we jump to doing in vivo.
But the point I'm making is you might decide not to do in vivo studies.
And I think that's a very good suggestion.
Be focusing first. Yeah, that's a good point.
We went and talked to Team Epidemics. They actually had a similar structure when they came into their proposal last year of an in vitro and followed by an in vivo. Their experts recommended to not do the in vivo and focus on the in vitro. And I think they'll tell you they're very happy they made that decision for monetary reasons, but also lab reasons. The amount of time in the lab with mice is very different than with cells. So you might want to talk to them.
Okay. You want to continue, or should we jump up to a library-focused question?
Maybe I can just add
Go ahead.
just one thing, because we're talking about the pH. So how many of you have had chemistry? Okay, so explain to me, the proposal here is you're going to add increasing concentrations of your bicarbonate. And you're hypothesizing that that's going to change the pH as a function of that concentration, is that correct? So somebody explain to me what a buffer is and how a buffer works, and whether I just want to know whether you have the right sort of thoughts on whether this is the correct approach or not.
Well, a buffer in general would be a weak acid or base. It doesn't dissolve completely in the liquid we're working with, and but since it is in our case, it is a base, and we've established that the tumor will have the most basic form of an acid, just free-floating protons, hydrogen ions in the medium. So the buffer, being a base, will tend to react with those hydrogen ions and take them out of solution. And since it is a weak base, not very many ions of that base will be dissolved, and those are the only ones that will be reacting. But once some of them are taken out of solution by reacting with the acid, that will disrupt the equilibrium of the system and pull more of the buffer to be dissolved, which will then react with more of the acid in that the tumor is producing. The more acidic the tumor microenvironment, the more acid is in the medium, the more of the buffer will be recruited to then dissolve and react with the acid and reestablish the equilibrium. So, yeah.
Okay. So you don't mention what the pH of your buffer is going to be. when you make up a bicarb, you have to make it up to a given pH. And so the answer that you've given me is correct up until a point where you saturate, where you're neutralizing the hydrogen ions produced by that tumor cell. And then eventually, it's going to stop being effective, right? You understand that? And so, the two things you have to consider with a buffer is the concentration of the buffer, but the pH of the buffer. What do you want this buffer to be able to, what pH do you want this buffer to be able to change it to? If you make a pH of 8, you're going to have the tumor microenvironment that's very alkaline, and bicarb actually works quite well at a pH of 8. If you make that buffer at pH 7.4, it's going to stop at pH 7.4 and not go any higher. 7.2 is the same thing. So you have to consider the pH of the buffer as well as the concentration of the buffer to give you your desired effect. If you put a buffer in there at pH 9, you're going to kill those tumor cells because eventually the tumor cells are going to be sitting at pH 9. They're going to say, where am I? So that concept of the buffer is important for you to think about. And so some people might just take a constant amount of buffer of bicarb and just change the pH of it. Okay, so if you have enough bicarb, you can do that experiment, and then you'll be able to actually measure the cytotoxicity at given pHs. And so you may want to think about that, adding that kind of complexity to your first aim.
Can I ask a very simplistic but I think practical question? Is volume of the added solution a concern?
I mean, the volume depends mostly on the concentration and pH, like Dr. Masper was saying. Because if you had a lot of volume at the same concentration, you'll get a lot more. But if you had less volume at a higher concentration, you have the same amount of bicarbonate.
I'm talking about delivery to the tumor environment. If you have to saturate the whole environment, you have to deliver at least enough volume to cover that cell mass, right?
Volume will be some.
Is that in a practical lab procedural sense, is that a limitation? Is volume I don't know, I'm asking a naive question. I don't know.
I feel like it's going to be a limitation because you can't inject you can't check entire body with 10 liters of bicarbonate or something crazy like that. But it's definitely in practical
Good big
sense.
mouse.
Nah, but it's definitely a limitation to consider as to how much you can actually inject, because sometimes it might become less poisonous to the mouse if it was in vivo and you put in too much and the blood became too diluted with the volume that you injected. So ideally we'd want to go for a higher concentration but in a smaller volume dose to get the same amount of bicarbonate without diluting the blood.
Just a thought.
I would add to that just by saying that while it is a targeted treatment, because it's being injected into a live model, there's a possibility for flow around the body and not just concentrate in that one area. So we would have to consider concentration. Some of what we inject might not actually affect the tumor.
Okay, good. All right.
I thought the targeted injection of the buffer was a good aspect of your proposal.
Okay. Should we go to the library side?
The only question I had is sort of related to data and the importance of data and collecting your data and deciding, and a lot of granting agencies, particularly in the health field, are not requiring that you have a data management plan before you start your work. So you do mention a little bit about having a lab note and having it written and then electronic, but I wondered if you could elaborate a little bit more on the system and start thinking about just what data you're gonna collect and how you're gonna store it.
In terms of the statistical analysis, I know you also had comments about that on our proposal as well. We haven't really hashed that out as much. We've kind of been working almost primarily on the background information and setting up how we're going to go about that. So I think that's one of our next steps coming up very soon. But it's good to know that a lot of grants right now want us to have a data management plan.
And it's because a lot of scientists don't think about it until
Right.
they get into
I think that's really important for us, especially with the limited time that we have,
Yeah.
to make sure we know exactly what we're doing before we start into that. But we haven't really quite delved into that very much.
Yeah, and that also comes up with the question, when I was looking at the methodology for testing in vitro, there's so many different assays and tests you can do and so much data you can get out of them.
Right.
And once you're reading it, you're like, oh my gosh, I want to do all of these because they're so interesting and they test for a lot of things, which is important that we need to consider what specific things we're testing and what data to get out of them. Yeah, that's a very
And as much as you can save that data too, then that can be shared along with your results, that other people might take your data and use it for other projects. So.
You'll want to also learn from team medics' mistakes. If you remember at Junior Colloquia last year,
Good
they talked
experiences.
about losing cells in media because of lab management protocol that they did not follow. So make sure you have a schedule. And it was a matter of Alice signed up to be there at 10 a.m. but showed up at 7 a.m. But Ben was there at 9 a.m. expecting to do what Alice already did because she didn't document it. So making sure that you have a schedule that you follow and that you stick to, and a communication plan, and also a way to record every step that you're doing. With so many people touching the cells, or if you end up doing the mouse model, working with the mice, you need to make sure you're documenting every piece so that you don't repeat a procedure.
Communication, when you're kind of doing research by committee, communication is very, very difficult. And a lot of times, the errors pop up. You have to be very, very careful whether you have a online shared document or whether you have a paper notebook that you sign in and sign out of. However you arrange to do it, you have to make sure that everyone sticks to the plan, whatever that plan is, so that no missteps are taken.
And building off of Eileen's question, how did your team explore, assess, and integrate scholarly resources into the proposal writing process?
APA formatting.
There's still some work to do on that.
Not quite there.
In terms of like researching and looking them all up, well, we split up a lot of our different sections, like the literature review and the methodology, into different subgroups based on subjects. So people would focus on immunology or focus on melanoma, and we all looked for our resources via that. And a lot of that was done through like databases through University of Maryland and the library, and just making sure we did run into the problem a little bit going through our resources when we were putting the proposal together, of making sure we had valid resources. And I think that's something we're definitely going to keep in the forefront of our minds going forward, is making sure as we go that each resource we're using is validated and is a true scholarly resource. Just because it ended up being fine, but we definitely had to like double check a couple of them, and I think that was a really important learning curve for us to make sure that the research we're doing, because we don't know a lot about the subject. So reading an article, like I might not know that the article is wrong in something it's saying, so making sure it's from a reputable source is important.
And just beware of federal agency reports that are reporting on works that others did.
Right.
And they're easy to cite because they're packed full of information, but you want to get that primary source that underlies that report.
And know too, if you have trouble finding resources, that you can come to me, and also with the data management plan, you can come to, there's resources out there to help you with that. So
And you all
feel free
did
to use me.
so you did a great job updating your proposal from the December version until now. It's much stronger and clearer, and you made a better argument for why your research is important. So watching how it has evolved has been rewarding, so well done.
And it will continue to evolve.
Yeah.
Yeah, I thought the proposal was really well written. I've got postdocs who had, Ivy League educations, graduate educations, and they have many more, typos and problems with their document than this one. So I think they're congratulated for that.
And I thought it flowed nicely too, it didn't.
They told a good story. Some of them are rough to read. This one told a story.
I'm gonna tell you how many times Dr. Shaw says, tell a story. That's like the number one thing, just tell a story.
That's
And we
kind of
really.
situation. I was not in good mood, so they worked very well. They were impressive because just less than 1 year, just 6 months stuff. They learned so much. So I'm really impressed.
Very good.
So
Some more questions?
No.
You good? Okay.
So you've got my comments from the,
Yeah, I forwarded them out to the
Yeah.
team, so we'll all take a look at them for our next meeting.
Okay, I would dwell for a moment on your budget for your project.
That was a lot of fun to write.
I know that it's kind of hard to write a budget for something you're just formulating how it's gonna work and you're just speculating, and we understand that. The whole purpose of this part of the exercise is for you to think about everything you do costs, and you have to find money to pay for it. So roughly a third to a half of your cost has to deal with taking care of the mice. Okay, so if you decide not to go with an in vivo phase at the end, yes, your lab work cost will go up, but not nearly what I think what mice cost will be. So that's something to think about. So you had a total budget right now, direct cost of about $5,700, which isn't bad for an experiment this complex, I don't think. How, where are you gonna get $5,700?
We actually,
Garrett.
my mom and I are friends.
Your mom's gonna write a check?
No,
Not quite there.
we're friends with someone who is in charge of it's called Kelly's Dream, it's a melanoma foundation in Maryland, and they're very good about fundraising, and we told her about our project, and she's extremely interested and wants to do a joint fundraiser. So we still have to work out the specifics, especially from a legal aspect, but what we want to do is put on a joint fundraiser with her, whether it be like a celebrity chef thing, or we have some contacts with some athletes and people like that we can get together for a speaker dinner and put together a joint fundraiser where her foundation would receive the money and then they would give us a grant in exchange for us helping them out. So we have to work out some of the legal aspects of that, but they want to meet with us in the next week or two and are very on board with that. So it's
That's fantastic.
pretty exciting, yeah.
Wear the same outfits.
You can't do that.
That's good. And remember the globs, if they want you to draw it, you know, the dinner napkin and say, draw this thing, remember the globs.
Yeah, artistic ability.
Okay, so that works out great. If you go down that route, talk to us before
Definitely.
you receive any money. There's different ways we have of receiving money from different sources.
And a gift is better than a grant.
We can, if the agency if the group wants to give you a gift, we can receive gifts.
Okay.
We also can receive grants, but then we have to route it through Aura and all that
Yeah.
kind of stuff.
And yet you get half the money.
We were all shocked by that when we
56%.
first learned about that. We were all like, what?
And we
Over
can
half.
also, if we talk to them ahead of time, before any cash change hands, we can arrange for, probably an indirect waiver for this group,
Yeah,
which is
that's
nice.
good, that's good.
So which means you get 100% of the money.
Would
Yeah.
one of you be interested in attending the meeting? I don't know if what your schedules are, but the meeting with the foundation, we have it?
What I would like for you to do, have the conversation as a team.
Okay,
If it goes to the next step, then we should be involved.
okay.
Okay,
Sounds
but
good.
lay the groundwork by yourself. This is part of the learning process.
Right.
Okay, so that doesn't go well. What's plan B?
Well, we've kind of put it on the back burner with the proposal coming up, but we have started to put together a list of grants that we are interested in applying to and trying to get money from. I don't have it in front of me, but
Okay.
we do have some there. We were planning on using some of the money we get straight from Gemstone to jumpstart some of the in vitro lab work and maybe some of our lab testing just right off the bat while we're waiting for those grants to.
We're gonna be sending out an internal request for proposals in the next 3 or 4 weeks. You guys open for that. It's not big money, but it's a little bit to get you started. Okay? Also, just remember, in general for grants, if you see a announcement that looks like it's up your alley and looks like they may be interested in funding you guys, either from a competitive research team or some strictly student-focused grant opportunities, if they have a deadline for submission, you have to have your grant ready a month before that. Okay, just move the date up a month, because I gotta see it, we gotta make sure it's going out here, then we have to route it in off campus, and just give yourself a month. So just pretend that real deadline doesn't exist and move it up a month, and we'll be in good shape. Your proposal reads real well. I mean, if you're gonna morph this into a proposal for funding somewhere else, it's a good place to start. You have to shrink it way down. But it's all, I think most of it's there. You have to tighten up your budget a little bit when you know exactly what you're looking for, but it's a good place to start. In pretty good shape. All right, that sounds all good.
So maybe I can add one more thing about the comment about in vivo studies. And the possibility of adding some defined mechanistic in vitro studies before you do those in vivo studies. It may be more palatable to a funding agency if you have those mechanistic studies and you say we're trying to understand a mechanism behind the anti-cancer effects here. That's a little bit more palatable to people than other than saying we 10 undergraduates are going to cure cancer. It's a little bit like when you get out of college, you get your first job, you're going to get an email that says, send us $10,000 now and we'll invest it and make you $100,000. And you say, well, should I just send this $10,000 off? Say, that sounds a little too good to be true. So, curing cancer is a little too good to be true, but understanding mechanisms is something that, that's a small step that could be very, very important. So I wouldn't I think for this proposal, you should definitely keep the in vivo studies in there, but I wouldn't make it the whole focus of the proposal and say, we're gonna try to understand how you can cure cancer. Really what you wanna try to understand is a small mechanism here, improving cytotoxicity by changing the pH. That is something that people could be very, very interested in.
Okay. Anything lingering on this end?
I think it's good.
And we always try to end up these sessions with having the team have the opportunity to ask us anything we may be able to help you out with. As far as questions, concerns, either from a very technical point of view or from a logistical point of view. So anything that's timeline oriented or process oriented, go ahead.
Just a question, going back to your first question to us about the in vitro study that you suggested. My biggest question in front of that is the T cells that we will be in vitro testing. From my background, I understand they should come from a mouse, but where would they come from?
So this is a T cell that originally came from a mouse, but it has now been immortalized. So you can grow boatloads of them in culture, and they all recognize the same antigen, which is derived from ovalbumin.
Okay.
So if you have a tumor cell that expresses ovalbumin, these cells will kill it. You just mix the two together and you measure how many cells die
Okay.
by whatever technique you want. You can, and so it's actually, not that difficult, an assay to do if you can figure out how to measure tumor cell death. Usually you label it with something, and when the stuff leaks out, you say, oh, that cell's dead. And so then you can really get a measure of this. But you can purchase these T cells or get them from someone who's using them, some cancer biologist who has them. Many people have these T cells. They've been immortalized and they have these weird names, OT1, OT2, DO1110, depending on whether you want to recognize MHC class 1 or MHC class 2. So you can get those cells, you can probably buy them, or you can get those cells from someone on campus here. I think maybe Dr. Fowler might have some.
We have that.
Oh,
Yeah.
And
That's
how,
why, based on that.
Free.
Yeah.
in
And
ter-
actually, so let me ask a little bit because you guys are looking at the tumor infiltrating cells. So these cells supposed to be coming from the tumor. So you need to, a small part, you need to have the mice, and then transfer it with OT1, which is Dr. Moser just said about, it's kind of transgenic T cells, and then put into the mice, and then you, kind of inoculate. And actually, it's not infect. I almost said infect, but it's inoculated the tumor cell line into that mice, and then the tumor will grow, and then the OT1 will go into that and become immune suppressive. And then after usually 15 days to 20 days, the tumor become big enough. You take them out, and then you homogenize and digest that and take the OT1 out of that, because the OT1 has experienced the immune suppression from the tumor. So now that's your job. You try to reprogram in vitro, use the IL-12 and B7 and the antigen stimulation, and also you try to change the pH in around the tumor, or in vitro or in vivo, and then to ask the question, can you improve the functionality of the tumor infiltrating OT1 cells, by in vitro system or eventually in vivo system. So it's kind of a little bit. That's why I said it's kind of complicated the system they choose. But that's I think you guys understand that very well so far.
and how translatable is this immortal T cell to actual cancer therapy? Like, it just seems to be manipulated so much that it's gone far from the reality of like an actual tumor and the actual T lymphocytes in a body. Or is that just
No,
my
I mean, you can get a lot of. You're correct in being, a little bit suspicious.
Yep, that's good.
But you can learn a lot from these mechanistic studies. It's an inconvenience that when you actually look into the tumor, a lot of the T cells that are in that tumor are non-specific or to other antigens. Many of them have nothing to do with the tumor; they just happen to be there. In this case, the OT1s that you get out of there are going to be antigen specific, so it makes your job easier, and it sets a precedent. So it's not an immediate jump to curing tumor, but it'll tell you a lot.
And how Like, what is the difference between MHC1 and MHC2 in terms of importance to antigen specificity? Like, is there a difference? Should we be focusing on both? Is the MHC1 more important than MHC2?
I think you're focusing on MHC1, and that's the right thing to do.
Okay.
And until you have immunology, you don't need to suffer the subtleties of the major histopathology.
But that's effective enough to look at, just the one, okay.
I'm gonna rewind you guys back 1 year. This time last year.
What was our
Diet
That's right.
and diet.
My
and
question, you have learned a lot since then, right? I wanna know, can I influence my susceptibility to cancer by changing my diet? To a more alkaline diet?
There's been a lot of studies out there. I've read the China study, and that talks a lot. I guess there, from what I understood of it, which is very limited, I'm not a science major, but I guess if you like, there are a lot of factors that are associated with. So a lot of what their study has shown is that like there are potential improvements in decreasing your risk of getting cancer, and in some cases, changing your diet can actually help the treatments and make it, I guess. It can actually like make the cancer go away, and that's kind of what I've read, but I haven't verified all of that.
And it was also really mixed too.
Yeah.
Like it depends on the person
I
a
think
lot.
diet is one of those things where it's gonna help you, but is it helping you because it's really changing the extracellular environment of the tumor? Or is it helping you because you're a healthier person in general, so your
Yeah.
immune system's
Yeah.
better off? And I know when I did research, a lot of what they had talked about was in osteoporosis, but the bone is very different than a cancer cell environment would be, and I think that was kind of where the flaw of that research question was. And like Al said, while it can help, I don't think on its own it would be a true treatment.
Actually, that is a very good question. I like the question. They try to say, hey, if it's not, because I question them, when you eat with acidic, diet, and then you change the tumor, so you have a long way to go from the digestion system to the tumor.
Yeah.
So how can you tell me that is true? So what we do is, they change it, so we do target change.
Yeah.
That's the reason, but that's a final, goal. If they can reach that, that's a very attractive idea. I like, that idea very much.
Well,
Similar
I don't think it would ever work. I have no data to back up my opinion.
My mom would be on the complete opposite side of what you said.
But it's a good idea. I like that idea.
All right, well, very good. You guys come a long way. I think you're on a very good path. It just, you gotta stay focused, stay targeted. If you think it's getting too broad, narrow it. Always narrow instead of expand. Those kind of things as you go forward. So, but good job.
I want to thank all of you guys for being here. Thank you for reading through our 60-page proposal. We really, really appreciate all of your feedback, and we'll definitely be incorporating it into our proposal.
Okay, great. Thank you.
Thank you.
Thank you.
All right.